It's 4 o'clock and I don't see a rush in numbers. There's one or so. So welcome to everybody to our webinar, SASSB webinar. And we're very happy that we have been able to secure somebody to come and tell us something nice, new and exciting. So Vandar Slenar will speak to us, but Kobus will introduce him in a minute. So just for the normal SASSB members, remember, soon you would get more information, but our conference will be this year here in Fabi on the 11th and 12th of September. So it's a Thursday and a Friday. We will have a workshop on phylogenomics starting on the Friday afternoon and the Saturday. And then on the Wednesday beforehand, we will have a workshop dealing with resources, all the kind of things, tools that you use. So if you have something special you can tell us about, please contact me. We're looking for people to help us and populate that workshop. But so keep the September open. We're trying to make it as cheap as possible. This is my mission for this year, is to make it as cheap as possible so that nobody can say they can't afford it. It will not be grand. That unfortunately comes with a price, but it will be good. So with that, over to Kobus to introduce our speaker.
Yeah, thanks, Vanus. So I'm quite excited for today. With Funda, we'll talk about species delimitation methods, and thank you, Funda, for being willing to do this for us. It's an incredibly powerful tool, and Funda has really been the person to bring this type of methods into Aspergillus and Penicillium taxonomy, for which I'm, of course, very grateful. But just a little bit more on Funda. He's currently a postdoc working in Prague at the Laboratory of Fungal Genetics and Metabolism at the Institute of Microbiology of the Czech Academy of Sciences. So he focuses on the systematics of filamentous fungi, especially for Aspergillus, and as I mentioned, pioneered the use of new delimitation methods for getting a better understanding of species relationships within Aspergillus, and it's led to several really key and important publications in this genus that simplified the taxonomy and identification of these important fungi. So Funda, we're really excited for your presentation, and I'll hand the floor over to you.
All right, so thank you very much, Kobus, for a nice introduction. And so I will get right into it. I'm going to talk mainly about species delimitation. Usually in my talks, I'm talking mainly about the fungi, and I'm just using the tools to explain some things within our studies about fungi. This time I will try to focus on the tools that I use for the species delimitation and for the taxonomic decision that we make in these studies. But of course, I will also touch a little bit on some issues that we are facing in taxonomy of fungi, and I will also try to show some things that are coming in the near future for the taxonomy, or at least I think they are coming. We will see. This graph on this slide, this is something that I like to use for the introduction of my talks. It shows the growing interest in the species delimitation topic and using these species delimitation tools over the past let's say 20 years. Of course, during this time, you can say that in general, the number of publication is rising in all areas. But here you can see specifically that in comparison with articles containing just the key term species. The articles with focus on species delimitation or using species delimitation methods is growing much faster. So this is just to show you that this is an interesting topic. And as Kobus said, I am mainly concentrating on Aspergillus, although we are doing, of course, studies or taxonomic revisions of other fungi as well. But the most of my studies focused on Aspergillus. So here is a very quick introduction to this genus. It's of course hugely important genus of filamentous fungi. It contains over 450 species, and the number is growing every year. The members of the genus are growing all over the world. They are used in various processes in biotechnology. They are of course acting as pathogens. They are causing infections and so on, producing mycotoxins. It's clear that it's just important genus. And here I wanted to start also with this slide, introducing the section restricti of Aspergillus, which the revision of this section was the topic of my master thesis. It was the first project that I started with, and it was kind of, I can say it was a blessing for me because it brought me to this species delimitation topic, because I got this section which was very understudied, little known. There were 7 described species, but the last taxonomic revision included only 12 strains. And I got my data set of 300 strains, and I had at that time absolutely no idea where should I start drawing the species boundaries. And I know that the simple phylogenetic methods wouldn't be enough to somehow finish this project, this revision. So I started digging into these species delimitation methods, and thanks to this, I learned a lot about them. So I will try to share some of this during the lecture. This is again to show you some interesting results of our work, of our revisions that we did in Aspergillus. We did more studies than these 3, but these 3 are nice examples of what we did and how we used these delimitation methods. So in case of the section restricti, during the study, it turned out that this is in fact an overlooked and understudied section, and within our data set we were able, or we were forced to increase the number of species within the section from 7 to 21. But in contrast to that, we also studied or did revisions of other parts of Aspergillus, like this series versicolores or nigri, and these are extensively studied parts of the genus. Even before our work, there were already a lot of known strains. The studies worked with higher number of strains, and there were actually quite a lot of species described within these sections. And based on our revisions, we decided that the actual number of species should be lowered quite rapidly, even mostly in the series versicolores, as you can see from 17 to 4. So this is the basic results of our work, and later I would like to show you how, or at least something of how we got to these results. Now, before I get to the challenges or issues of the taxonomy of fungi, I would just like to mention the species problem, as it's called, the question about the actual existence of species, or if not existence, then at least the proper way how to define them, how to define the species category. So I learned during my studies that this is actually much more active level of research that I actually wasn't aware of before, and I think many people are not that you know, it's not that known that this is still like unresolved issue, and I just mentioned several recent publications which are really interesting, and they get really deep into this problematic, into this topic, and I just recommend them to anyone working in taxonomy to understand, at least to some extent, that there is also this level of problems in taxonomy. So it's nicely demonstrated by the fact that there are at least 32 different species concepts in taxonomy across across all organisms, and it's also interesting to note that taxonomists they usually are convinced that the species category is actually real, but the higher level classification is probably artificial and is not present in the nature itself. So this is just a very short introduction to this topic, but I believe that it's really the fact that we are not entirely sure about the existence and the proper way of defining the species category. It's hugely influencing the way that we work in taxonomy, and when we are trying to define the species, that delimitate the species, even though there is not a clear definition about what the species actually is. So now to the more specific issues regarding the fungal taxonomy. There are, of course, many of them. There could be just lecture discussing all these interesting topics, but at the bottom of this slide, I list some of the issues that I don't want to really discuss because they are not directly connected to the species delimitation topic. Of course, everything is of course connected to everything, but not as directly. So, for example, difficult life cycles of fungi, they are complicating taxonomy. Dual nomenclature, of course, complicating taxonomy. Hybridization, that's very interesting topic and understudied in fungi. But again, I believe it's not directly connected to the stuff that I will discuss later. But the other issues, the other topics mentioned here, I believe they can be clustered into these two sets of issues. So the first set is concerning the taxonomic characters or taxonomically informative characters like phenotypic or specifically morphological data. This is of course problematic in fungi because there is not that many morphological characters that we can rely on in taxonomy, and at the same time lots of these traits or characteristics are plastic within the species or within population or whatever. so they can change a lot if you are able to assemble large enough data set. I will show some results of our work regarding this issue. And another connected topic to this is the acceptance or rise of cryptic species. I will talk maybe several other times about cryptic species during the presentation. So again, this is also directly connected to the previous problem that I was talking about. So again, how are we deciding on whether something is a cryptic species or not, if we are not entirely sure about the definition of the species category? So this is definitely interesting interesting idea and interesting topic for discussion and for more studies concerning just this topic specifically. And the second cluster is concerning the sampling issues with fungi, and by this I mean the fact that if we want to sample specific species or specific section of some genus or even genus, it's not straightforward task with fungi, mainly with microscopic, but I think with fungi in general, it's not just it's not just basically it's not possible to just go to the nature and try to sample specific species. So this is of course again complicating the taxonomic work and the species delimitation work in particular. And there are two other connected issues connected with this sampling problem, and it's the fact that there are lots of unculturable taxa which are sampled only through metagenomics or DNA and environmental DNA sequencing, and also the fact that there are these estimates of the total number of fungi. So I will get to these topics a little bit later. So first of all, this is the example of the morphological plasticity even within the genus Aspergillus, where again this is a kind of topic that it's not which is not as known in, I guess. So I wanted to demonstrate here. These box plots show the measurements of several morphological traits of Aspergillus series versicolores, and the colors are showing different species. And I just wanted to show here that the variability within the species, even if they are very narrowly defined, which would be the shades of these colors, like the blue and red, then even within the narrowly defined species, there are often very, very large differences in the measurements of these micromorphological traits. And these differences are often exceeding the variability which is present in the whole series. So not only we are having low number of taxonomically usable traits, but at the same time they are variable within the species. And what's even more, or adding more to this problem, is the fact that often in taxonomic studies there is very limited number of strains that are examined in this way. So in many, I would say, in many studies, you don't even realize that this is actually an issue. So this is just to show that this can be something to think of while conducting some taxonomic revision. Then this slide is here to discuss a little bit more the cryptic species. Again, since we are not entirely sure how the species category should be really defined, and we always, if we are studying the species boundary like really close enough with lots of data and lots of individual samples, strains, then we will start to see these gray zones between the two species, and then the decision whether or where should we draw the line between the species, if some amount of differences between two species is already enough or not, is getting arbitrary. And this is getting a little bit more difficult with the, I mean, I'm not trying to say that cryptic species do not exist. Of course they do. There are many reasonable examples, but it's just complicating the taxonomic decisions if you are like adding this to all the other issues that we already have. So then moving on to the actual species concepts which are used in fungi. Again, I am using the example of Aspergillus, but I believe that this issue or this treatment is quite similar across other fungi, at least those that are like a little bit more thoroughly studied, but it applies basically to all species, genera, or taxa in general. As I said, in Aspergillus, there is a golden standard of using polyphasic approach to taxonomy, which means collecting as many data from all these different types of information as possible, and then combine them all for the taxonomic decisions. But since there are all these other issues with the taxonomy, it often results in DNA data, molecular data, or phylogenetic trees being used with more weight than the other data. So of course, integrative taxonomy, of course, it's the goal. We should try to get there, but the fact from the recent studies, I would say, is that the phylogenetic data phylogenetic reconstructions are getting more importance in the eventual taxonomic decisions. So moving on to the estimates of the total number of fungal species. So why is this a problem? I would say it's because the actual number of accepted species in fungi is somewhere around 150,000, but the estimates of all the fungal species are moving from something like 1 million to 10 millions, and these are the, well, I think I can say conservative estimates. There are even studies estimating higher numbers. So this means that there is just a very small proportion of the fungi already known, and this is, in my opinion, it's influencing the way that fungal taxonomy is moving, as we are, or the general idea is that we are just trying to find as many species as possible as quickly as possible, because in fact there is so many of them that we are not even closing to the final number. So here I just demonstrated with this phenomenon with these numbers that there are twice as many species described per year now compared to 25 years ago. And now to the sampling issues, which is something that I'm really concerned with. And again, I believe it's something that it's not as much discussed as it should be. It's my opinion, but I think when I tried to search for more literature on this topic, it's difficult because it's not as much discussed as it should be, in my opinion. Because for the, and again, I will probably get back to this throughout my presentation. It's really important for the species delimitation methods. It's always mentioned that it's important to have intraspecific variability within the data set, and it's hard to obtain in fungi, and as a result these are numbers get together by Cobus that again, in Aspergillus as an example, we can see that there are more than 50% of the species are known from one or two specimens. So this within species variability is really missing, and I think that this is really widespread in fungi, and also the fact that these species are still described and accepted is, in my opinion, again possible because of the reliance on molecular data and phylogenetic trees. Yeah, I already mentioned series versicolores, and this was, I'm really happy with this study, with this revision that we did, because it's rather small part of the genus, but it's extensively studied, and we had very large set of strains for such a small group. So I think then we were able to delimit the species boundaries with much more confidence than is usual due to the sampling issues. And as I already said, we decided that the number of the species should be reduced, because when we get to this level of sampling, we saw that the other way of dealing with the revision would be describing like extremely high number of species, which would be really unreasonable. There would be no morphological or phenotypic basis for describing all these species. So in the end, it really only made sense to reduce the number of species. Okay, yeah, as I said, another connected topic, of course, is the large number of unculturable taxa. These taxa which are known from the sequence data, but they don't have any physical samples. They are called the dark taxa. But I think this issue is also related to these more studied and more known genera or taxa in general, like Aspergillus, because there are, for example, certain parts of the genus which are not entirely unculturable, but much more difficult to work with, difficult to cultivate. So again, this was the example of section restricti, which was understudied because the species or the members of the of this section are xerophilic or osmophilic. So they in nature they are growing in dry conditions or in conditions with high concentration of osmotically active substances like sugar or salt. So they are not as frequently observed in classic mycological studies. So again, it's just that this issue again is wider than generally accepted, and it's connected to these other issues. So that was about the fungal taxonomy issues, and I think that it's just all of these issues are for me they are leading us to the way that the phylogenetic data or molecular data and phylogenetic reconstructions are very important, even probably even more in fungi than in other organisms, where, for example, the sampling issues are not as frequent or as widespread as in fungi, and therefore we are more relying on the molecular data and phylogenetic reconstruction, and therefore we should employ these species delimitation methods, at least to make the taxonomic decision more robust, more valid, like this. So that this was just my introduction why I think that the species delimitation methods are really important in fungi. So now I will introduce you this species delimitation workflow that we used in our studies, with some some changes, some advancements over the years. In every paper we are changing it a little bit, but we are always adhering to this basic workflow pipeline or something like that. So, and it's based on this like really influential paper published already in 2013 by Carstens, and it divides the and also this pipeline. It's not always. There are, of course, many studies employing species delimitation methods, but they are, for example, using just a fraction of the methods, or maybe just one of the tools for the whole study. And of course, it's okay as well. But there are many papers which are citing this as the proper way to do the analysis. So the analysis is divided into 3 steps. Often the first step is called species delimitation, which in my opinion it's unfortunate because the whole analysis is of course species delimitation. So there should be some other way to describe this first step, and I like to use this term or this phrase, creation of species hypothesis, but it's not usually used in other studies other than mine or ours. Then the second step is inference of the species tree, and the final step is species validation. So now I will go through each of these steps in more detail. So the first step, the creation of species hypothesis, there are quite a lot of single locus methods, meaning that they are created in such a way that they use as a input some either alignment or already phylogenetic tree reconstructed based on single gene data, and the results of these methods are usually used as the starting point for the analysis, and then analyzed further. But sometimes because there are so many different studies taking a little bit different approaches, you can also find some studies which are using, for example, current taxonomy, like the currently accepted species of the studied genus or taxon or whatever, or they are using the phenotypic data as this starting point instead of these species delimitation methods. But in our studies, or again, usually in fungi, it's already this first step is based on molecular data, and one of the tools that I really like, and because it's, first of all, it's multi-locus method, so you can use more molecular data at the same time, and also over the years, over the different studies, I observed that this method is really producing like meaningful results that were that were agreeing with our our idea about what we saw from the other data, like the morphological data and other methods and so on. So this is just it's because in the paper that I mentioned, the general recommendation is to use as many methods as possible. So it's a little bit in the way of integrative taxonomy, even on this smaller scale. So no method should be treated as better than other. It's always best practice to use as many methods as possible. Compare the results. Use the consensus. But I think if I should choose just one method to go with, I would choose this one of these available methods. But the important and interesting fact about this method is that it's hugely influenced, or the results of these methods are influenced by one. There are many parameters that you have to set while preparing the run of the analysis, but in most cases it's just fine if you follow the manual to the method, use the default values. But this one parameter is really important to. It's important to set it correctly, but it's difficult to come up with some meaningful values. Again, it's more difficult in fungi because this parameter is based on the effective population size of your species and on the mutation rate, and these are values which are I guess it's possible to come up with some meaningful values even in fungi, but it's difficult. So what's usually done, not only in our studies but in general, is that there are used multiple values of this parameter, and then the different results of these different values are compared. On this slide, I just wanted to show you the typical representation of the results of this first part of the analysis. So you can see the colored bars on the right. They are showing the results of these different analyses for different loci, and the different colors shows the delimitation into separate species by each method. So you know, when there are incongruencies in the delimitations among the different methods, it's represented by these bars going further, or you know, being split into separate colors if this method separated this data set based on this gene into more species. So back to Stacy. As I said, it's a typical practice to use several different values of the collapse height parameter, and this is the representation of the results. So you get your data set, and then these boxes in the shades of gray, they show you the delimited classification or delimitation of your samples into the hypothetical species. So if you use higher collapse height value, it will cluster more samples into less species. So higher collapse height value, you will get less delimited species in your data set. And here it's you can see two different values, two different results. On the right hand side, the samples are clustered into less species represented by these black boxes. So what we did, or what I wanted to do from the beginning, but actually get to do it a little bit later during my research, not in the first paper, but it took me several studies to get to this level. So I decided to use a large number of collapse height values and explore the influence of this parameter on the results, on the delimitation. So this graph is the result of this exploratory analysis, and it shows you with the growing number or the growing value of collapse height, you get this curve which shows you the number of delimited species, and it's getting lower. And at the same time, it shows you the support for the first and second most probable scenario is the result of the species delimitation at the certain parameter value. So with the growing value of collapse height, we are getting less species, and the different scenarios, different delimitations are getting higher and higher support. But what's important is that the biologically meaningful values of the collapse height should be probably very low, very small, probably as small that we would get no like none of the scenarios would be supported at all. So my idea was to try to search for the lowest possible value of the collapse height parameter that would get us some at least reasonably supported scenario. So in this case, we can argue about this peak down here at the low level, but then there are of course these two large peaks on the right side of the graph, and then we get these. We can get more of these scenarios and then compare them with the during the species validation step. I decided to include also this graph, this picture in the presentation, and this one is not based on the real data, but I wanted to show you here to make it more clear. I hope in my mind it's getting a little bit clearer. So this is based on simulated data, because the real data would be too messy. But this is showing you not the first and second most probable scenario, but this is showing the distribution of several different scenarios with 4 delimited species, 6 delimited species, 10, 16, 20, and it shows you that at the low collapse height value you are getting these scenarios with many delimited species, but with very little support, small support, and over to the right you are getting less delimited species but higher support. Okay, so sorry. So moving on to the second step of the analysis is this one is quite straightforward. This is called the species tree estimation, and there is more or less just one tool that is used almost always, and it's called StarBeast, and it just takes all of your samples, and you have to decide. Again, you can do this in several different ways, several different settings, because you decide in this case on the species boundaries based on the step 1, and it will combine more samples into one tip representing the species that was delimited in the first step, and then you can see you can visualize the results with this program called density, and it will nicely show you the incongruencies in your data set with these conflicting parts where the support is not clear for the topology of the tree. Okay, so this is, as I said, this is rather straightforward. And then the last part, the species validation, it's again slightly more complicated, I would say. So there are two programs or tools which are widely used, mainly the BPP. It's really widely used, and it's interesting tool which can combine actually all three steps of the workflow. Or you can use just some of the steps. You can use it just for the validation, just for the species tree estimation, or just for the delimitation. But again, it's largely influenced by values of some parameters, like this mentioned ancestral population size of the species and the divergence time of the species. And again, these are hard to estimate for our fungal species. So again, this is very interesting tool, but it's difficult to set it up correctly, and maybe that's why I actually prefer the second one. And there is also some discussion because they are based on slightly different assumptions and so on. But the other tool, delineate, it's influenced only by the tree that you use as input, and as input you are using the tree that you calculate here in the second step. So to make it a little bit more clean, hopefully I showed this concrete example of delineate analysis. So you get this species tree for your data set, and then you can, or you have to, fix some of the types, some of the lineages in your tree, and you leave some of the lineages unassigned, left to be delimited by the software. In this case, they are highlighted by this yellow color, and the only limitation that comes with this tool is the high usage of computational memory. So you have to think about the way that you will calculate your tree in the second step, so that you will not end up with many lineages that have to be left unassigned, because if you use too many unassigned lineages, the analysis will have troubles like finishing properly. But otherwise, I really like this approach because you can create the species hypothesis in the first step, then you create the species tree in the second step, and then you use this species tree in this species validation step, and you can test different scenarios that you create in step one. So what I then actually present in the studies is this kind of representation or graph, or whatever, that shows several different models that we tested within the validation step. And in this case, the gray bars, they show the fixed lineages, which are the software is sort of teaching itself based on these fixed parts of the tree that you decide to fix for the software. And then the brown bars, they represent the unassigned lineages that will be delimited. And we use these red frames around the brown and gray bars to show the results of the validation step. So the red frames will or are showing the decisions made by the delineate software. So that's it. That was like quick, but just at least a little bit deep overview of this workflow that I use. And here are just summarized some of the advantages that I see in this approach. So it offers you an unbiased look at your studied group. If it's used properly, it should enhance the clarity and objectivity of the taxonomic decisions. It should result in consistent species being delimited over, for example, your genus that you are studying, or possibly even among the large taxonomic groups. And it's also universal because you can use it with, it's not tied to specific markers. You can use it with whatever is available within your studied system. Of course, it comes with some challenges, or again some issues. So most important, what I always say is the within species variability. It's mentioned in almost every manual of all these methods always, that you should have variability within species. But at the same time, it also means that you are not, you shouldn't use identical samples in terms of your data. So the samples might not actually be identical, but in terms of the data you are using, they can be identical, and then it might cause problems with these methods. Also, still there might be problems with accessibility, because I understand that it's still not as easy to use as it should be, but I mention here some or this effort which I really like and support. It's called iTaxoTools, and it's trying to make these tools more accessible for people which are not, for example, happy with using the command line and so on. So it's making these tools available, and it should enhance also the reproducibility of the research and so on. But one more thing that I see often in the studies doing this species delimitation methodology is, for example, using the multi-locus trees within the single locus methods framework, which is also influencing the results very much. And I decided to mention here this. It's quite old study, already 25 years old, but it's even before, like, I guess all of these methods were developed, but it was already discussing the delimitation of species based on multi-locus data already at that time. Even this specific paper was even dealing with fungi, and it was discussing that even without the tools, methods, programs, you can do it based on just phylogenetic trees of these different loci that you have in the multi-locus dataset. But you shouldn't just use the multi-locus tree. On the other hand, or in contrast to that, you should use the single locus tree and look for the incongruences within the data set, and right at this point where the incongruences, of course this is very simplified way. There are many processes of evolution which are complicating this view. But this is basic, but it makes sense, and I like this idea that right at the place where the incongruencies between the different loci appears, it's the point where we should probably expect the limits of the species, and it's just, I just discussed this because I think it's a shame when people are they do have the multilocus data set and they use it wrongly because they are just they create the multilocus tree and use it with some of these single locus species delimitation methods. So it's just to mention one issue then, or challenge that can come up with this species delimitation framework. And now I would like to, I'm getting to the end of the talk, but I would like to discuss a little bit some interesting things that are coming in the future and that will influence the delimitation of species, specifically in fungi, and that are again touching on some of these issues that I discussed at the beginning of the talk. So of course it's the whole genome data. It's practically already happening. It's not future. And again, I will discuss this quickly one by one later. This is just the introduction of these topics. Then the this topic of genomes used as types, which is currently discussed in fungal taxonomy. Then, of course, the employment of machine learning for species delimitation, and also the advent of metagenomics, which is also kind of already here, but at so far only at the amplicon level and not the whole genome level of metagenomics, which is probably also coming in some future, hopefully not not so distant. And this multilocus based species delimitation, which I was talking about in this presentation, and that we used and we are going to use it also in the coming studies, it's kind of getting out of date already, but I believe that it's still it still has its place in taxonomy. And yeah, I will show you one more example. So just to say a little bit more about the whole genome data. So I just checked that there are already more than 20,000 whole genome sequences of fungi in GenBank, which is, I would say, pretty high number, but also it's a little bit more complicated by the fact that there are certain species that are overrepresented within this set. So it's not like there are 21,000 different fungal species. It's, I don't know the exact number, but it will be much lower because there are so many important species which are sequenced over and over. Of course, the top of the list is Saccharomyces cerevisiae. And one more thing that I wanted to mention in this context again is or are the cryptic species, because with the whole genome data, it's quite easy to get some variability, which you can then present as interspecific variability, or that the differences are large enough. There are enough of them to you know, pronounce this strain as new species or different species from the other closely related. but then we are getting into this area of possible taxonomic inflation, describing too many sort of meaningless species. So again, it's just whole genome data are it's great, it's fantastic that the data are coming, growing, and everything, but we have to be a little bit careful about using of them. Again, it's crucial to obtain the intraspecific variability within our data sets that we are dealing with, which again, it's getting, of course, easier because the whole genome sequencing is getting cheaper and cheaper, but still it's something that is, so far it's problematic because there are not that many whole genome data, I would say. Of course, with the growing number of genomes, this is also important and much discussed topic. Genomes as types. I mentioned 2 studies here which are discussing this topic from the positive way and the negative way. There are of course many arguments for both sides, and I think that in my opinion it's coming in at some point, but maybe we are not there yet. We definitely the rules have to be decided really carefully to make it useful and not just a tool to create many new names based on potential artifacts in the data. So of course it's a difficult topic, and I understand that there is a large resistance within the community, but on the other hand, there are of course many advantages of using genomes as type. So I'm just it's going to be interesting if this is going to happen at some point. Machine learning for species delimitation, I really enjoyed one of the previous seminars, which was discussing exactly this, and I think again the combination of machine learning and whole genome data, it makes perfect sense because the traditional methods are not really capable of dealing with the amount of the data that is currently available, but machine learning is like in contrast, it needs these large amounts of data. And in fact, I would say that I am rather surprised that there aren't already much more studies using machine learning for species delimitation. I think it must come at some point, but so far there are not as I said, not as many studies as I would expect at this point, because it's already I don't know, maybe already 10 years, no, maybe not yet 10 years, but something like that, since the first studies using machine learning for delimitation was published. So maybe again it's about the accessibility, because probably the tools have to get a little bit easier to be used by higher number of biologists. Maybe there has to be some review article which would again set some basic rules for using machine learning. Maybe one other issue that might limit the use is that machine learning so far, as far as I know, there are not these models of evolution which are available for the classical model-based not taxonomy but phylogeny. But I think at some point it's also coming. So maybe some people are waiting for this, but I think definitely this is the future, and we will see much more studies using machine learning in coming years. And also metagenomics. It's in some ways, again, it's already here. There is this very interesting resource called Global Fungi database, which is using large number of metagenomic studies, and it's really, this is changing the way of this is directly influencing the issue with the limited sampling or problematic sampling of fungi, because even for some I use this example of Aspergillus restrictus. Otherwise, it's problematic to sample this species, but here there are already more than 1,000 samples from almost all over the world of this species. But of course, the problem or not problem, but limitation possibly so far of this database is the use of very short DNA sequences. It's not mostly, it's not even the whole ITS region. It's just, for example, the ITS2 sequence, which is like 200 bases or maybe even less. It's not really ready to be used in species delimitation studies. But if we get to the point that we will have such kind of database, but with whole metagenomes assembled, then this would like change everything that we do so far in terms of species delimitation. So I'm looking forward to it. Yeah, I'm getting to the end. So I would like to finish with this study that we do together with Cobus, where we are still using the classic species delimitation methods that I discussed today, but again we expanded the scale. So this is the example of series Veridicata of Penicillium, and again this is rather small part of the genus, just 8 currently accepted species, but we acquired large number of strains, large number of whole genome sequences, and this picture right here, it represents the preliminary results of the species delimitation methods, and it's based on 61 genes which are based on this universal fungal core genes, which were decided recently in this paper. So yeah, I mean, I wanted to demonstrate that this classic model-based species delimitation, although it's not maybe the state-of-the-art approach already, but it's still valid, still has its place. If it's used correctly, it has this huge potential to increase the taxonomic stability possibly over the whole not only fungi, but I mean all organisms. So that's it. So I would like to thank all of my collaborators, of course, all of my funding. I mentioned just some of the collaborators, but yeah, that's it. Thank you so much.
Thank you very much, Vanda. It's always great to listen to you, and I'm sure there's a lot of questions, and I certainly have a lot of questions. I spent a month with Vanda last year, where I asked a lot of questions and learned how to do these methods. So I'm a little bit worried that he says that this is already old-fashioned methods, but I would say still very valuable. Before I ask questions, I can maybe, Vanda, if you can just maybe unshare your screen.
Yes.
Then if any of our participants have questions, you can just put up your hand and we can take some questions. Farnes, who's a bacteriologist, where you were speaking about what our future prospects for fungi is, he just showed that they have all the tick boxes.
For the bacteria.
So I did mention that now you just have to start using coalescent approaches as well to make sense of all of your data.
We try.
Yeah, I also saw these interesting studies that were discussing the species level in organisms that are not reproducing sexually, and again it's not resolved, it's not finished topic, but I think there are studies that are showing that the species might actually work pretty similar in the asexual asexual organisms as it is in the sexual organisms. So it's interesting that these methods that were developed maybe mainly for, I don't know, animals or something like that, that it might work in other realms of organisms as well, even in bacteria and so on.
Yeah. Hendrik, do you maybe want to ask the question?
Yeah, you can just unmute yourself and ask the question.
Okay, not a problem. I thank you very much for a very nice talk. I really enjoyed all the methods that you guys are using in order to delimit these species. It's always a big challenge. I'm not from the fungal world. I'm more of a botanist, and I know you mentioned that you really don't want to talk about hybridization and those sorts of things, but I do note that it is probably still a big part of fungal evolution, hybridization, and I wondered if in that pipeline of yours, incorporating some sort of a test for hybridization might not also be prudent, since the coalescence-based methods like Stacy or some of the other ones only really model incomplete lineage sorting as the cause of the incongruence, and might it not be prudent to also look for that sort of things, especially since in the fungal world, I suppose, hybridization can sometimes lead to some really nasty things, especially in Aspergillus, for instance.
Yeah, I think it's interesting that you are mentioning it, because I myself also I'm convinced that hybridization is very important in fungi in general. That it's like largely understudied topic, and we are now working on hybridization in Aspergillus, and I think it's mostly matter of not the hybridization not being really easily recognized in fungi, because you know it's really you cannot see it, of course, based on the morphological methods. You cannot see it, of course, by if you are sequencing one locus like ITS, the general barcode, then it tells you nothing about hybridization. If you use multi-locus data sets, you are mostly still getting nothing, and then only if you get to the genomic level, it's possible for you to find something, but I think you have to look quite hard to find the marks of hybridization in the genomic data. So there are already several published Aspergillus hybrids, but it's really so far there are very little or very few cases. But in case of Saccharomyces and these related species, which are much more studied, there are much more genomic data of them. So there are already some known species which originated by hybridization and so on. So again, I didn't want to discuss it really. It's probably it might also influence it definitely influences the species delimitation pipelines, but you really have to have the correct data to be able to find some marks of hybridization. So yeah, that's it.
Fantastic. I just since you also mentioned the cryptic taxon thing and the cryptic species, I'm thinking it might result if there's more genomic data coming out, you might eventually decide to unsync some of those things that you might have sunk in your pipeline now again, when you might realize some of those morphological differences were maybe linked to some sort of a trait situation. But I definitely understand hybridization is a messy business. And definitely still one of the biggest challenges that at least us in the botany world are struggling with. I'm sure everyone else who's working with any sort of hybridization really does struggle quite with it. But thank you very much for a fantastic talk, and I really enjoyed that.
Thank you.
Yeah, I think the issue with fungi, I think there's only Aspergillus latus that we know all the genomes coming out is a duplicate with another species. So in those cases, it's quite easy to at least fish out the data from the genomes. But sure, yeah, I think we've been ignoring hybrids thus far because it's so complicated for us to interpret, basically.
Yeah, exactly. And it's even because you really have to look for the hybridization, because if you just get the genome and get some typical markers, then you might miss the fact that something is in fact hybrid. So yeah, but I mean it's nice that there is this example of Aspergillus latus, so that we know that it's possible. And I hope, because last year on IMC there were several other people presenting posters with Aspergillus hybrids, that they are producing them in vitro similar to us. So again, I think more data or more studies regarding hybridization in Aspergillus is coming in future.
So yeah, thanks for that. I was just thinking, because fungi has a great skill to explain things and make it seem really easy. And so I thought maybe you can just talk to what's the learning curve to learn how to actually do these methods. And you kind of hinted towards the fact that you think, Stacey, if you had to learn one of these methods, Stacey is one of them. But I thought maybe you can just give some perspective as to, how long do you think it takes to actually learn how to do these methods, and then to run a data set? What's the time frame that you're looking at?
Yeah, I mean, I'm afraid that maybe during the talk, I maybe made it seem a little bit more complicated than it actually is. But I would say it's for me, what's nice and what I like about it is that there are different levels of complexity, and I'm still learning, and I guess I will be learning for as long as I would want to. So you can always dig deeper. But if you want some like really basic pipeline, really want just to include it at the smallest possible like cost, like time cost or whatever, it's I would say it's already pretty easy with these as I mentioned, these iTaxo tools. It's really beautiful effort. When it was first published a few years ago, I was a little bit afraid if they will develop it, but they are maybe it's not like very fast. It's not growing to some huge amount, but it's growing. They are adding stuff, and I would say if someone just want to include some basic level of species delimitation in their taxonomic study, the initial amount of effort it's really not, it's not that hard. It's getting pretty easy. So I would encourage everyone to just try it for themselves.
Yeah, that's great news. And I think that we've been trying to get Funda to come to South Africa as well to do a workshop, but we'll keep trying to get you on a plane down to Pretoria. Do anybody else have a question for Funda? Otherwise, I guess we can wrap up.
Well, I mean, thank you so much, and yeah, I want to just say at the end. Still, I spent several years trying to learn about these things, and I don't feel like an expert. I'm always afraid that some of these people, like for example, some of the authors of the methods will find out some of my, studies or something that I say here, and they will say, "Oh, what is he saying?" So always just, I mean, check everything again for yourself to be sure. But I mean, I really enjoy learning about these tools, and it's nice to try to move it a little bit to add something to these topics. And it was great that I could share some of it with you, and thank you very much, Kobus, because it was I learned as well. I learned a lot last year when we met in Prague. So thank you.
Thanks for that. Yeah, and I'm thinking in the worst case scenario, you point the people who are developing the tools towards some Aspergillus literature, to get some culture. But thanks a lot, Funda, and thanks for driving, incorporating these methods in these important genera. And I really do believe that it sets an example for the rest of fungi and other groups to also follow. So with that, I'll close down. Faunus, I don't know if you want to say something.
No, I just wanted to say thank you very much for the time and effort, and for everybody that joined us. Thank you very much for listening. We want to make systematics available to everybody. That's what the society wants to do. So thank you for that, and that we can share these kind of things. So have a good evening, everybody. Enjoy your summer. We are dealing with our winter. All the best. Bye.
Thank you so much. It was a pleasure. Bye.
Ja.
So. Nou,
Ja,
Ik heb al zijn meisje, dat is bijna.
ik heb gezien mensen.
Dat.